Hey, it's Mr. Lum, and we're going to be talking about more about DNA, RNA, and protein synthesis. So hopefully you have a copy of the guided notes. Um, and yeah, anyways, let's get started. Okay, so first off, what are proteins? Proteins are these large uh, molecules made up of what are called amino acids. Um, Now, amino acids all have a very same structure. They all have what is called this amino group right here, this NH3, or sorry, NH2 group, and they also have an acid group right here. Okay, and then they have a central carbon and a hydrogen. The R part, so first off, let me just back up and say all amino acids have this chunk inside of them. That is the base. And then there are 20 different amino acids, and this R just represents that's where the variability happens. That means every every amino acid will have this chunk right here. And then some amino acids will have what's called an R group. Now, as the body builds amino acids, or sorry, as the body builds proteins, it they stick together. So here's a bunch of amino acids, and you can see that each amino acid sticks together. Now, how do two amino acids stick together? Well, the amino group is technically a base, as in like, and this is an acid. And acids and bases want to react together. And there's a whole process that you'll go into more when you go into like biology 12 or something like that. But if I were to draw two amino acids together, like here's H, H, and carbon, hydrogen, the R group, which is everything else, then C, O, OH. So that's my one amino acid. And then here's my other amino acid. And I'll have like, I'm just going to draw half of it. So there's that carbon, that hydrogen, that R group. Then I'll draw my amine group. N, H, and then another H like that. So what happens is that these things right here combine, this O, this H, and this H, and they actually fly away. And what does O, H, and H make? Well, hey, that's funny enough, it makes H2O. And so these things combine, and that base and that acid want to combine, and it kind of separates, and then it makes an H2O, leaves it, and it's called a dehydration synthesis because it loses the water. And yeah, and that's how amino acids stick together. So amino acids are like right here, they start to stick together, and then they get folded into what is then called a protein. So, and we're going to learn a little bit about that. Before we do, so what do proteins do? So proteins perform functions in cells. They can be structural, like fingernails and hair, which is like keratin. They can be like structures in the face, which is like collagen. They can be for like transporting hemoglobin. Hemoglobin, or heme, is one protein, and then globin is another protein, and that's the main component for blood, for red blood cells. And transporting like oxygen and stuff like that through the body. They can also make different kind of chemicals like insulin, things like that. They can do receptors on cells to like absorb water for a cell, to help you absorb food and sugars for cells. They can also be defensive in nature, making antibodies and they can also be making muscles. And a lot of people think of this one right here is proteins make muscles, which is actin and myosin proteins, which do contraction, and cells that do contraction. And of course, the last one is they can also be enzymes, which speed up chemical reactions. So this is an example of lactase. This is an enzyme right here, lactase, and it helps you digest lactose, which is milk sugar, or one of the main components in milk. And it's a protein that helps you digest sugars. Here's just kind of like a video of like things kind of being built along the way. But yeah, so again, proteins will build all of these different things. Now, there's a couple of different structures to proteins. So first off, when a protein is being built, it starts off as just loose amino acids, and it then those amino acids get stuck together and it forms a chain. We call that chain a first degree structure, or sometimes we call it a primary structure. First degree or primary means the same thing. So amino acids form a chain. Then afterwards, it forms a second degree structure or a secondary structure. And what happens is this chain right here twists and it turns into this, okay? Then it turns into a third degree structure, or folds into like a 3D shape. And that's this right here. So imagine you had like a chain. This is like a physical, like metal chain link chain, okay? And say if you started like twisting the chain around, right? Eventually it's going to start to coil up and kind of make a little bit tighter pack, like this secondary structure, okay? And say if you kept on twisting and twisting, it's going to start to fold in on itself, and it turns into this tertiary structure. Now, what a quaternary structure is, or a fourth degree structure, is when you take a bunch of these tertiary structures and you stick them together. So this is actually, looks like it's like 3 or 4 different kind of proteins that are here. Well, not proteins, but 3 or 4 different tertiary structures. So here's one tertiary structure here. Here's another one. And then we have that other big one in the back right there. And all of these different kind of things come together and they make a quaternary structure. And this is what your protein is. It'll be in the shape of something that can transport. It could be hemoglobin. It could be lactase to help you digest milk. But that's the process. The first 3 things aren't proteins. Until it gets into that last structure, that's when it becomes a protein. Okay, the analogy is kind of like, hey, a primary structure is kind of just like random letters, okay? And then it folds and it starts to turn into words. And then you have a tertiary structure, which is kind of like making a sentence. And then a quaternary structure is when you link a bunch of those sentences together and it can make like a paragraph. So how do we make a protein? We talked about this before. It's through a process what is called transcription. DNA gets read, and then since it gets read, it gets then copied, and that is called transcription. It transcribes, or in other words, rewrites the DNA and rewrites it as RNA. And it stands for, and it's mRNA, messenger RNA, which is the same as what the vaccine is made out of. But this is how all proteins are made in the body. So DNA gets transcribed, that means rewritten, but it rewrites it in a slightly different way. it writes it as RNA. So it just uses, like, again, we talked about some of the differences of RNA and DNA. Then that RNA gets translated by a ribosome. So a ribosome reads this messenger RNA, translates it, and brings in a bunch of amino acids and turns it into a protein. Actually, first turns into a primary structure, and that primary structure twists on itself, turns into a secondary, twists more, tertiary, combines with other tertiaries, and forms a quaternary. So where does this all occur? The DNA remains in the nucleus, and then the mRNA is in the nucleus, and then the mRNA leaves the nucleus, and it then goes to the ribosome, and that's where the ribosome assembles the protein. And here's just an example of that. Like, you can have a gene on a flower, which is just DNA, makes a protein, and then that can get exported out to, be color or whatever it is. Okay, what are some of the differences between DNA and RNA? Now, we talked a little bit about the differences yesterday. One is that DNA is double-stranded. RNA is a single strand. We also talked about the different type of sugar that it uses. DNA uses deoxyribose. That's the sugar it uses. And we talked about deoxy means it's missing an oxygen. And we can see that deoxy is missing an oxygen and ribose has that oxygen. So that's the difference between, and that's, since it has that different type of sugar, that's why it gets a different type of name right there. There's one difference that we didn't really list, and that is DNA uses T, C, A, and G. RNA doesn't like to use thymine. Instead, it uses a different base instead of thymine. It's very similar to thymine, like looks very similar, but it's called uracil. And here's the example. This is a picture of thymine. This one right here is a picture of uracil. They combine very well. Thymine always combined with adenine here, okay? And since uracil looks so much like thymine, adenine only combines with uracil when it's in RNA. So these are the main differences between DNA and RNA. Thymine is switched to uracil. They're single-stranded, and the sugar is ribose. Ribose has one more oxygen than deoxyribose. Now, there are three kinds of RNA. There's messenger RNA, which carries the code for the protein to synthesize to ribosomes. Messenger RNA is the copy from DNA. Okay. There's rRNA, or ribosomal RNA, combines with protein to form a ribosome. Now, how ribosomes work? Ribosome is a organelle in the cell, and how ribosomes work is, and how this messenger RNA sticks to a ribosome, is that they stick by with all that rRNA, that ribosomal RNA. And the final type of RNA is what's called transfer RNA. This little thing right here is the amino acid. And what happens is that this part right here is transfer RNA. So this could be, let's imagine that this is uracil, okay, which is like thymine. This is cytosine. And let's imagine that this is guanine, G right here. So U, C, and G right there, okay? Well, this U, C, and G wants to combine with this messenger RNA right here. So that could be like A, G, and C. And these three things combine with this, and it drops off this amino acid. And then the next three will combine with a different transfer RNA, which is attached to a different amino acid, and that's how a protein is built. So there's two steps to protein synthesis. Transcription, which is the DNA being changed into RNA. That RNA goes out of the cell, and then the ribosome translates that messenger RNA and turns into a protein, and that's what we call translation. So transcription is DNA turns into mRNA, and then translation is mRNA turns into a protein. Now, we're going to kind of skip over this little part a little bit, but I don't want you to focus too much on it. But DNA is copied into a small strand of RNA that can. Now, what translation is mRNA brings code to ribosomes. What we mentioned before, ribosomes read 3 mRNA bases at a time and puts an appropriate amino acid in place. And these 3 base sets are called codons. Now, as the ribosome reads the messenger RNA, transfer RNA brings in the amino acid. So again, here's your messenger RNA. Now, these things right here are transfer RNA. Okay, they combine, as you can see right there, they combine and they bring in these amino acids. Now, each transfer RNA has its anticodon that base pairs with the codon, and then corresponding amino acid at the other end. So again, here's the codon, which is right here. Now, the part that is on the transfer RNA is this part right here, which is called the anticodon. Okay, the anticodon is the opposite of the codon, or in other words, combines with it. And the amino acids are held together by peptide bonds, forming a polypeptide. And we talked about what a peptide bond is earlier. A peptide bond means that water leaves, and it allows 2 amino acids to stick together. Okay, so transfer RNA continues to bring in amino acids until a stop codon is read. At the very end of messenger RNA is a stop codon, so that the ribosome knows that's the end of building a protein. It knows that we're not going to add any more amino acids to this protein. Then translation stops and the polypeptide is released. The polypeptide is a fancy way of saying essentially the protein, or the polypeptide is a fancy way of saying a first degree structure. It's the chain of amino acids. So this is just kind of like a thing that says, here's your translation, brings in the amino acids, transfer RNA with the anticodons, and combining with the codons on the messenger RNA. Okay. Now, like I said, there's 20 different possible amino acids used to make proteins. And when you have there are 64 possible combinations of codons. And this leads to a lot of duplicates, okay? And so this is a safety mechanism in your body in case DNA hypothetically accidentally gets changed. So, for example, let's imagine that a little bit of DNA gets changed, and therefore messenger RNA gets changed. And so here's your messenger RNA. Let's imagine that a good messenger RNA will be GUU, like that. So guanine, uracil, uracil. And something changes in that, and some radiation occurs. You're no longer GUU, you're GUC. Okay? Well, both GUU and GUC code for the same amino acid, so have no problems. And same thing for this one. Like, this is if UUA changed to UUG or vice versa. That's LEU stands for leucine. It's an amino acid, and it's not going to be a big deal. So let's figure out, can you determine the corresponding amino acids to the RNA codons? So what we have right here is, I give you a codon, and we're going to figure out what amino acid is associated with this. So I have right here CAC. So we're going to look up CAC, and we find CAC on here, right there. Okay, and so it makes that codon. Now, these are just like abbreviations of all these. Like I said, like leucine is so like Leu is short for leucine for that one. But yeah, and HIS stands for histidine, but you can just write HIS for it. Okay. Now let's find these other ones right here. So we have GUA, and we have GUA right here. That's Val. Okay, and hopefully you were finding the rest of them. But again, Val stands for valine, which is another amino acid. GCU, and we find that GCU is right there, alanine. And we have AAG, which is AG is lysine right there. Oh, no, sorry, not AAG, AGG is, yeah. Well, this is arginine right there. So now what I want you to do is, here's a DNA triplet. I'll do the very first one, and I want you to do this worksheet right here, and you're going to finish this one off. But I'll do the very first one right here that says TAC, and you have this in your guided notes. It says, so TAC, that's the DNA. So now we need to figure out what is the codon for that. Okay. So what combines with T? Well, it's going to be A. A. What combines with A? it's going to be T. Oh wait, there is no T's in RNA, there's U's. Okay, what combines with C is G. Okay, so now we have AUG. That produces a amino, that produces this one right here, AUG. And what codon is that? That is the start codon. So you have a start codon and you also have stop codons. So often you will see start codons. So this, what codon is that? That's actually not, or sorry, what amino acid that? That isn't amino acid. That's start. Like that. Okay. Now I want you to finish the rest of these. You can put the video on pause for a little bit, and you can finish the rest of these, and then we're going to move on. I also want you to try to finish these things. It's the same kind of thing. A small error has occurred. I want you to try to figure that one out.